Okay, and now we can have our speakers join the stage for our considerations of gene therapy panel. This will be led by Ms. Emma James. Do you mind if I announce who's up here? Okay, this includes Dr. Bradbury, Dr. Borsani, Dr. Lee, Dr. Guo, and Dr. Gray.
Anyone that's ever seen me do this will know that I don't ever get up on stage without water, and yet there's nowhere that it fits. Okay. I'm so sorry. Is that everyone? Well, good morning, everybody. For those of you who don't know me, my name's Emma James. I work for Encoded Therapeutics, but that's not why I'm here today. Just by way of introduction of Encoded, we're a gene therapy company developing gene therapies for CNS disorders. I have been working in the gene therapy space for over a decade now, not just at Encoded. And I work very closely with the Combined Brain Group. I chair their industry advisory board, and that's really how I've come to know Amber. We've become friends, and of course, as you all know, once Amber knows you and she identifies you, she pulls you into the SLC6A1 community. So I'm very pleased to be here today. We had some really, really exciting preclinical data and clinical data presented today. I guess just by way of starting, could one of you please sort of talk about your thoughts about how we might go about thinking about translating some of this preclinical data into human studies, and what some of those challenges might be.
Yeah, no, I mean, look, I think this is like with any drug, it's the balance of safety and efficacy, and I think that we've seen some data today that we know that we can overexpress SLC6A1, or we can express the wrong cells, and we can drive toxicity. And I think pretty significant toxicity, including animals dying. So we need to be really, really cautious about this approach, and just make sure that we're keeping that in mind, and we're any of the studies trying to model like dosing in paradigms that would really match how we're applying this to humans, and just making sure that the vectors we're using are extrapolating to humans the way that we think they are. We're talking about some of these next generation capsids, and making sure that we understand how those doses in animals relate to doses in humans, so that we don't sort of come into a situation where we know that there's a toxic threshold and maybe the vector works better in a human.
Yeah.
And then and that might, that could have been a situation where there was a next generation capsid, that Capsida had a clinical trial for STXBP1 where the first patient died apparently within a week. And that could be one of those possibilities.
Yeah.
So I think that's just, I know we want to move fast, but we know from the data, from the studies we've done, that we need to be cautious.
Yeah, I think I will just add, I just totally agree with Steve, and I think to further de-risk our gene therapy, I think is very important. One thing that we have been doing is actually also looking at human cells. So we've heard from many speakers today, and we're using our own strategy, but different promoters, different capsids, and so on. One thing I think we will need to go back to the patients is the human relevance, the genomic relevance. The promoter that we are using should be active in human cells. And we using mouse as a model, and which might be great, and we see the mouse can live, can walk very nicely, but is it working the same way when we put the same virus in human and patients? And we're also talking about the genetic background of different patients might be different too. So there are many of those variants that we might need to test explicitly. So I think to further de-risk that will be very important.
Can I just put you on the spot a little bit? Going to Steve's point about the Capsida situation, just in case the community hadn't heard that the STXBP1 community recently had a child treated with a gene therapy with an alternative capsid, and sadly that patient died following that. So I obviously the alternative capsid work is incredibly exciting. In terms of the ability to potentially treat these CNS disorders with intravenous capsids. But how do you think about the shift in risk-benefit for the patient population, given that?
Right, so this is really timely and really acutely aware of that. This is a situation is still ongoing, is still progressing. When this happened, I think the field is like, okay, we can take a stop and just step back and see what's going on. And we don't know much about how the patient passed, and I think there are more to be revealed in the later months, if not years. So hopefully we'll know a little bit better what's going on in that. We actually contacted with Capsida as one of our potential partner, because from our mouse work we know that a very broad targeted brain barrier penetrable capsid will be useful. So Capsida is one of those on the market, but not the only one. For example, Apertura, that's the one that the other one could be considering as a competitive, if you will. They also have their own brain barrier penetrable capsid. So would that be a better version? We don't know. We need to test it. But I think there are. The field is still moving. And for example, Steve's AAV9 fire, it's on fire. So it could be great. It could be a great option for us too. So that's something we want to open for options, and we are agnostic to what vector. We will choose the best one simply. Okay. But this is something still we need to be acutely aware, and we want to be cautious but optimistic.
Okay. And I just wanted to go back, and sorry, and then I won't just speak to the two of you, but I wanted to go back to one of the comments you made about safety. You mentioned the ability to increase potency by between 3 and 10 times, which again is very exciting in terms of the ability to potentially dose with fewer viral particles. But do you think that increased potency carries any potential safety concerns with it that we need to be aware of?
Well, so keep in mind, it's the idea is you're targeting 3 times more cells. It's increasing the number of cells. So you can do that by either increasing the potency of the vector or increasing the particle dose. So by having a vector that's more potent, it means, okay, if there is toxicity, well, you could lower the dose to a point where it's still effective, but you're at a lower dose, which makes it cheaper to manufacture. Reduces any of these risks with having a higher sort of capsid particle dose. So you always want a vector that is more potent. Just like you'd want any drug that is more potent, because then you can just use a lower dose.
Anyone else have anything you would like to say to any of these?
I agree with what's been said, but as far as your translation to the clinic and potential barriers or considerations, I think the capsid, the route of administration, and the dose are the real 3 big things you have to consider. So in our instance, we felt that AAV9 was there's so much safety data on it that's kind of tried and true. In the Capsida example, there are new and better things, but they clearly come with increased risk. And even if non-human primate toxicology studies are performed, a mouse and a monkey are still not humans, and there's really no way to predict how those viruses are going to perform in the context of a human patient.
Yeah, that was going to be a great segue to how do we think about again that translation. So you've all presented some great data in animal models, mostly in mice and rats. Obviously, the brain of the mice is very different to the complexity of the brain of the NHPs, which is very different in terms of the complexity of the brain for humans. And we can't really study biodistribution in humans because that would require taking brain biopsies. So how do you think about, again, how we translate between those models and then predict the approach to actually going into the humans.
So I feel like doing NHP study is kind of essential for part of the translation, just considering how the vectors will respond to the cells, the transduction and the spread. Because for mouse, it's easy for us to get brain-wide expression. It's exceptional, and we target over 30% of the cells with just one capsid. But in primates, that's a totally different story. And that's the reason of the different transduction and spread. You may have lower efficacy, and you may have to increase the dose just for that, and that increase the safety issues. Yeah. And then a lot of the question we're also facing with NHP study, because NHPs are normal, healthy animals, yeah. Right? For GET1, potentially that's actually limiting ourselves, because they will be able to tolerate even less drugs because they don't want additional GET1s in their brain, right? But all the patients are heterozygous, they actually need the gene. Yeah. So it's kind of a dilemma. We want to test the spread, but it's actually kind of another safety here because we can't go over the superficial homological levels of it.
Yeah. Yeah, I guess just for everyone here to understand, so typically when we're predicting human doses for humans, we look at safety and efficacy in mice, and then we use those data to predict a safe dose and in NHPs, but because we don't typically have actual models of the disease in the NHPs, we can't actually test for efficacy. And so then we use the data both from the mice and the NHPs to figure out a safe and potentially efficacious dose to go into humans. Before I carry on, does anyone in the audience have any questions for the panel?
What if it's a next generation capsid that can be tested in primates as well?
Just in case everyone didn't hear that, what if Amber asked for them? Yeah. What if it's a next generation capsid that cannot be well tested in NHPs?
Yeah, look, this is one of the big. So the next frontier of all of this is these next generation capsids. I would say AAV9 is what we've been using for the last decade because it's what we have, and I'd categorize it as most of the time barely good enough. And so these next generation capsids are going to open things up where we're going to be able to deliver the gene much better across the whole brain. We're going to be able to use much lower doses. They'll be cheaper to manufacture. Like, this will get solved, and this is the future. But with that, the problem is a lot of these capsids that people have developed only work in monkeys, and they don't work in mice. And so you can't test them in the mouse model of the disease. Or the one that Henry mentioned, this Apertura capsid that targets the human transferrin receptor. It's very, very specific for the human receptor. It doesn't work in mice, it doesn't work in monkeys. So they've made humanized mice that have the human receptor, and it works beautifully in them, like real game changer, like just knock your socks off good. But it's a leap of faith saying, we've got these mice that have the human receptor, it works really, really well in those mice. And we're going to have to go straight from that to humans. So it should work. It's the same receptor. It should work, but we're not really going to know until the first human is dosed. But I think with all of this, one other thing that I just want to bring up is I think that we've done a good job, for the most part, of being very transparent about these various efforts that are going forward. We're sharing information. We're sharing data, and this has been a huge step, actually dosing a patient. And I know Emily, talked about can't really share data on the clinical trial until it's further along, and these data lock policies and stuff. But I think that it's really important to be as transparent as you can possibly be about what's been done, especially the safety signals. And and I guess there's a comment that so far the safety has been good, but I didn't see. Can you share what the dose was? Was this a single-stranded or a self-complementary AAV? This was self-com? Okay, I was adding up the elements. It looks like it's too big. 6E13 vector genomes was the dose. Okay. So that's almost 20 times lower than what a lot of other trials are dosing right now, like lumbar intrathecal, AAV9.
Yeah, it's comparable to one of the Batten's trial doses. It's comparable to one of the Batten trial doses, and because we did have that toxicity signal that we've all talked about, we really had to go in with a dose that we felt confident that our IND-enabling tox studies covered the dose range.
Add something. Yeah, so just want to add something on what Steve talked about, the humanized mouse model. So this is actually the challenge for that, because the virus does not work in a mouse or in non-human primate. So that's why the strategy will be using a humanized. What we mean is that the transferrin receptor that the AAV is working on is humanized. So we swap it in the human version of it. And like Steve said, it works beautifully. How we translate to clinic, then we need to design a bridging study using a tool capsid that has the equivalent brain-wide expression virus, but work in mice, and then use that as a bridging study to compare the two strain and the virus. So that's one way we try to bridge that, and of course that's something is a real faith, and we will need to check on that. The other point I want to add is that we're really limited by the animal model to test the efficacy, like the mouse that we use in heterozygous, which is more relevant for disease situation. It's actually not showing much of a behavior problem. What we can detect is the EEG spike, which is great. But we don't have a very conspicuous phenotype on that. So that's a little bit different from the human situation when you have heterozygous in patients that are all heterozygous, but you have seen all this developmental delay and whatnot. So a better model will be necessary. One model that we have not talked about, we have not seen so far, is human organoids. So these are the up-and-coming cell culture model, but they are mimicking the early brain development and miniature brain, if you will. And then those are human cells, but they are made into this what we call the organoids, and we can grow in the lab. And testing our gene therapy construct in the organoids might be another way to complement the study that we cannot do that in mouse. But of course, human organoids, they don't have behavior, they don't show, so there are limitations of that also. But I think this combinatorial approach might be necessary for a better de-risking of our potential therapy.
This was a great session. Thank you. I'm Jean Paz from UCSF. I've been coming to these meetings thanks to Amber for many years, and I want to say that a few years ago it would have been unbelievable that treatments can work at the later stage of the development. So I want to say how excited I am to hear that. We heard several talks, but we also heard that there might be side effects. So my question is, to what extent are the side effects or the adverse effects, right, caused by too much SLC6A1 expression versus the immune system? Thanks.
Who wants to take that?
I mean, I can only speak for the mouse models, and I think for our studies, most of the side effects like the seizures, the mortality and spikes likely are linked with the physiological levels of GAT1. Basically, the brain is really sensitive of how much GABA transporter you have. You don't want to put in too much, then you lose GABA. But I think with the human trials and also the NHP study, it's a combination of both, right? I mentioned earlier, the NHP are normal wild-type animals. They have the immune response from the vector. Also, they have the add-on of the additional GAT1 transporter. So together, they may have a really complicated adverse effect.
We've talked a lot about, sorry, did you want to answer?
Well, no, I was just going to say separating like the AAV toxicity and immune responses from the here. I think we're worried about GAT1 overexpression. I think that we've got a pretty good body of knowledge, a pretty good understanding of how to, what any toxicities around AAV itself would be, and how to manage those immune responses. So I think the focus here is really on GAT1 regulation.
So perhaps I'll ask you to just expand on that a little in terms of the AAV, because everyone today has really talked about AAV-based gene therapies. Has there been any work done using either alternative viral vectors or perhaps non-viral vectors? And if so, why or why not?
I'm just going to say that I think that there's a lot of exciting technologies that are kind of under development, and there's data being presented on different approaches using synthetic nanoparticles or possibly other viral vectors. But right now, nothing that I'm aware of is kind of ready for prime time. I think AAV is still the vector.
Can you repeat that question again?
So fundamentally, they become mosaic. So some are expressing, some aren't expressing, some are hyper-expressing.
Hold on, Ryan. If you're going to talk that smart, you need a microphone.
So fundamentally, you're making them mosaic. So you have some that aren't expressing, some that are expressing a lot, some that are expressing a little. It's tapering off over time. So you've made mosaic, and you've taken away the ability for clinicians to reach back for pharmacophore treatment, because they are expressing. So now you can't, oh, I'm going to do this or that. So how do you think about that? How do you address that?
Well, my stupid answer is we need to get it. We need to. So my stupid answer is that we need to get the gene therapy efficient enough, effective enough, that we're treating a high enough percentage of cells that's managing the disease reasonably well. That's my stupid answer. The reality is that's a tall order. So don't have a good answer for you.
I think I would try to unpack this question to different levels of how that mosaicism is. I think if I understand correctly, the virus once goes to the system, it will have a quite sustainable expression, and it could be years, if not decades, right? So then we can consider that as a different status, if you will, right? Ultimately, what we want to do is to have an increase of that GABA transporter back to where they should belong. So functionally, if you can restore, then this reversibility should kick in, right? So that's what we found in our mice.
Yeah, but his concern is that you're going to have some cells that have this level of GAT1, other cells that have low levels of GAT1, and so if you're trying to treat it pharmacologically, you're affecting all the cells up or down the same way, and so it might be that creating problems in the treated cells when you're trying to overcompensate, right? Yeah.
I can see Scott's waiting for a question.
Yeah, I was just going to comment on the aspect of not having viable pharmacologic approaches post this mosaicism scenario. And I don't think that's quite accurate how I would think about it as a clinician. Many of the drugs that we are using don't act in the GABA system at all. Even the ones that we do, it's not as precise as this. If our pharmacology was that precise, then we would actually be treating the disease, right? So I think there still is lots of pharmacologic options available to be able to treat symptomatically after a gene therapy.
So I know we are almost at time.
Okay, one more.
What?
We're just gonna have two more.
Okay.
Thanks. Hi.
Amber says.
Hi, Alex, Boston Children's Hospital. Can anybody imagine a tool or heard of one that would let us image how much tissue has been transfected in a human? In other words, a leap of faith to estimate how many cells out of the total cells available are going to carry the repair gene. And in medicine, if something doesn't work, it's only for one or two reasons. It's ineffective, or there's not enough of it. And eventually we'll come to a point where in some humans we'll have efficacy and some we won't. Is anybody that you know, I don't, working on an imaging technique or maybe a molecular method, or maybe some subtle biopsy protocol, or something along those lines, to identify how effective was the gene therapy in a patient, not from a clinical perspective, but how many cells actually received the payload.
That's what we need you for, Alex. So that's what we need you for.
So the only thing that I'm aware of is there have been some instances where there's been some post-mortem analysis of some patients that have received gene therapy and unfortunately passed away. We had one of those happen in our clinical trial for giant axonal neuropathy, that there was a comprehensive and rapid autopsy. We did a biodistribution analysis on that patient. And so they got a dose of 3.5 e to the 13 AAV9 intrathecal, and based on the vector genome copy numbers, it was at or below 1% of cells across the brain that would have received the gene.
Hi, I'm Kelly Kang from Banbury University. So this is very exciting. I have a couple questions for the panel. So do you think all the patients with different remaining function will receive a same flat dose? This is my first question. Second question is, can same patient get redosed? For example, if a patient receive maybe dosage feels too low, in the future, can we redose? Also, Henry mentioned about a specific time window. So what's the speculation? So why do you think only work in certain timeline? How do you
So essentially, do we think everyone's getting the same dose, and can patients be redosed? Essentially, yeah.
I think it's going to be really, really challenging to try to tailor doses to specific alleles, specific mutations. You have to have a lot of supporting preclinical data, and to sort of suss that out for every different allele is going to be challenging. As far as the redosing, the dogma right now is that you can't redose with gene therapy. Patients are essentially immunized after the first treatment, but with at least the intrathecal gene therapy, Novartis just published a paper showing that basically they could dose monkeys that had actually really high neutralizing antibodies against AAV9, and they could still get almost unaltered distribution of the gene throughout the CNS. So I think that opens the possibility that you could have redosing protocols, and a lot of people are working on that. Because a lot of people want to do that, but right now it's not typically done.
His vector, if we only work in certain time window, maybe his can have a redose, maybe your fire is on fire all the time.
There's lots of cross-reactivity of the antibodies. I think that's not.
And I'm aware that there are some immune evasive version of AAV that is being developed right now. So that may be in the future, and when those AAV that can evade our immune system, then maybe redosing is a possibility. But right now, I don't think this is something possible to do that. And in terms of the window, I think it's related to the brain development, and when I wear my hat of developmental neurobiology, there are critical periods where those GABAergic circuits are maturing, and then they will consolidate. So those are what we call the critical period windows, where once it's finished, it's difficult to rewind those circuits. So I think that might be part of the process that you limit a therapeutic window successfully. Maybe there are some pharmacology that we can rewind those circuits, those reopening the critical period, but those are the next generation or like the future research that can tell us.
So I know we are over time, but I want to pose one more question to the panel. So Amber at the beginning said patients and families are begging for treatment. So how do you think about, or how would you suggest families think about taking the therapies that they have available now versus waiting for options that are further down the road?
I don't mean to be the only one talking here, but look, every family has to make their own choice here based on their own situation. gene therapy is permanent. I will say that there are orders of magnitude better technologies that are like on the horizon, where I think we're going to be able to, it's going to be much easier to treat this disease with gene therapy. I think we're doing what we can right now, and I know Allison and Emily had, pretty significant constraints, safety issues, where they couldn't dose this as high as, as they would have wanted to, that I think we all would have wanted to. And they had to focus on safety first. But I will say, because the stakes are too high here, Amber's talking about, expanding the trial, enrolling additional patients. That dose is, you're going to be getting single-digit percentages of cells targeted in the brain. It needs to be a higher dose. If we couldn't dose it higher, because then we need to design a better construct that regulates the gene better so that we can dose higher. So every family needs to make the individual choices. I'm hopeful. I really hope that things work out here. But I think it's possible to do a lot better.
Anything else?
If I may add, I would just end it on maybe a high note, and like one of the speakers said, the future is bright. When I read the literature about GAT1 knockout, the mouse is available 20 years ago, and there are lots of work that's been done in California. But at that time, we were studying the mouse. Now we talk about patients, we talk about clinical translation, and Steve done a lot of great work on that, and there's so many programs. We have a lot of experience on that. So we're just really following the path and try to navigate that. And I think, it's becoming a reality, and we have not thought about that even 10 years ago, 20 years ago, but now this is a reality. So I think that's very exciting. As a bench scientist, this is something going to clinic, this is very meaningful and fulfilling.
Yeah, I would agree. I mean, just hearing all of the amazing science today, I'm blown away by how many people are researching this. So I do think it's an incredible time. And it's just, everyone needs to really focus on understanding what the available options are. But again, I think, the future's brighter today than it's ever been. Yeah? Yeah, of course. Totally. Yeah.
Well, thank you, Amber. I do recognize some of Steve's concerns, and I do also recognize that the field of gene therapy is advancing rapidly, and there's always going to be new technology on the horizon. And it's been a long 5 years, and when I was putting together my timeline of development, it was both therapeutic and anxiety-ridden at the same time. Thinking about just all the studies that it took to get here, and to obtaining the Maxwell mouse and making 5 iterations of a gene therapy, and then performing dose escalation studies and safety studies, not only in mice, but we talked about primates. We actually chose a different route and used pigs due to cost restraints, and non-human primates are actually difficult to get. A lot of them are actually seropositive for AAV, so we used an alternate large animal in pigs. It took a lot of regulatory interactions with the FDA. A lot of people think the FDA is scary, but they're actually here for guidance, and we really appreciated being able to work with the FDA throughout this process to help us select a dose that was both safe and what we really do hope that will have benefit. We wouldn't go into a patient if we didn't think that our therapy was going to have benefit. That would not be ethical. But the first phase of every trial is safety. So first is do no harm. We want to make sure that we're not doing harm to our patients. And we really do hope and have glimmers that there's going to be therapeutic benefit. And so it's been really, really rewarding. This is the ultimate reward for what we do. We come to the lab every day. We give up time with our own children and our own families, and spend countless hours on the bench and with mice and writing grants to fund this work. So the ultimate reward for the work that we do is to actually see something translate from the bench to the bedside, and I'm just really happy to be part of this SLC6A1 community and partnering with Amber and all of you. Thank you.
Thank you. Thank you, Tanya. So, Amber? Are we sticking to the time and coming back? Yeah. Yeah, no, I mean, we're wrapping up. This is over, but when should everyone come back?
Hello. So lunch is downstairs in conference dining. And what we're going to ask is if you could go and maybe get your lunch and bring it up, or just to get us back and keep us on track for the afternoon. So there's snacks and beverages out there, and something will be out all day. But as far as lunch goes, if you could go down, get your lunch, and get back as quickly as possible so we can keep going. So thanks so much.
Thank you for having me.